Good afternoon, everyone. My name is Juan Castaneda, and I'm a rising junior at Tufts University. And this summer, I had the pleasure of working with Dr. Lutton, and we studied antigen presentation in the little skate. So what is antigen presentation? It's an immune process, therefore it's very important for us to understand the two branches of the immune system. So we have the innate and the adaptive immune system. And just a very quick overview of both. The innate immune system is the first response to an infection by a foreign pathogen. The adaptive immune system is a little bit different because it can tailor the molecules and cells that it uses so it can be specific to that foreign invasion. And even though they're different, something very important has to occur between both of them for this response to actually occur. And that's for them to communicate. And this happens through antigen presentation. So this is a schematic that will show you how antigen presentation occurs at a physical level. So you have the antigen presenting cell from the innate immune system that can engulf that foreign entity and chop it up with its internal machinery. What it can then do is take bits and pieces of that antigen and present it on the surface of the cell through a receptor called MHC, or major histocompatibility complex. And you can think of it as a pair of hands that's just holding out the antigen, waiting for something else to come grab it. And that other pair of hands are the TCRs, or T cell receptors. And those are located on the surface of T cells. And this binding has to be very precise. It can't be too loose or too strong. Otherwise, the T cell is not able to mature and carry out some of its intended functions. And I want to bring to your attention this actual interaction. While this is a 2D representation, this occurs in a 3D world. So these structures are very complex, and that will play an important role in how I set up my experiment. But it's important to address my actual model organism, the little skate. Why do we use the skate in my study? So little skate is part of the elasmobranch subclass of cartilaginous fish. So what does that mean? So essentially, it and its relatives, the sharks and rays, are the first time in an evolutionary timescale where we see the expression of these MHCs and TCRs, which are so crucial in antigen presentation. Additionally, they're really useful in the lab because they are cartilaginous fish. Therefore, their immune organs aren't enclosed by bone. So if we want to study bone marrow in mammals, we actually have to degrade the bone, and we risk degrading some of the cellular samples we want to use in our studies. And the little skate actually has a lot of the organs we do. For example, the spleen is another immune organ they share that we have, and we know that antigen presentation occurs in that organ. They also have some very unique ones, such as the Leydig organ, and that's found in the esophagus of the skate. And what's really interesting is that in the literature, it's always been thought of as the site of leukopoiesis, or the birth of these blood cells. However, back in 2013, one of Braun's students, Jesse Lupica, made a really interesting observation and a finding in the histological analysis. So what we're looking at here is the inner lining of the esophagus and the muscle mass that it's enclosed by. And we see what looks like a tapeworm being engulfed by the epithelium into this space, and they create a pathogen trapping compartment. The second photo demonstrates, and the resolution on the slide isn't too well, but it's better on the computer, but believe me when I say there's innate immune cells here that Jesse was able to identify. And it's really interesting to see that those cells that really start the antigen presentation process to be so close to the Leydig organ. These are all Leydig cells around this site. So it's really puzzling to see the first few steps of antigen presentation occurring near an organ that has been previously thought not to have that quality. And so I asked the question, can the Leydig be the site of antigen presentation? And the way I address that question is by saying that if the Leydig can act as the site of antigen presentation, then an infection will induce expression of those molecules, the MHCs and TCRs, that are so crucial in that process. So how did I choose my genes of interest? So I want to bring you back to the receptors that I've actually been using or talking about. And I want to remind you that they're 3D structures and they're made out of multiple parts. They're not just one receptor that's made by one unit. They need various parts to come together to be functional. So I've gone ahead and boxed together the units that work as the receptors. And so those are 8 out of the 10 genes I'm interested in studying. I'm also looking at these recombination activating genes because they're the ones responsible for the creation of these TCRs and what allows them to be so variable in an organism. But I did say I wanted to study this in the context of an infection. So last year, 2 students, Marina McKinnon and Alex Dimmond, started a parasite inoculation study. So what they did is they isolated this pathogen, Calliobrothum versitulatum, from the gut of the hermit crab. And the hermit crab is a natural prey of the skate. So what Marina was able to do is isolate the pathogen in culture, which then allowed Alex to administer the pathogens to the skates through 2 different avenues. One was oral administration, and the second one was through an intraperitoneal injection, so pretty much through the bowel cavity, to guarantee that the skates receive the pathogen. So we had various different time points in which we retrieved the organs we're interested in studying, because we know that immune responses don't occur at one particular time point. They're very dynamic, and they can occur during an extended period of time. So for every time point, we had 3 animals, 2 experimentals that received the pathogen, and 1 control animal that simply received a ringer solution. So I step in, and now I'm doing the molecular work of the project. And I was really lucky that I was able to come in, and tissue collection was already done, so I had what I wanted to start. And my focus was really in primer design, but I want to take you quickly through the first few things I did during my first 4 to 5 weeks here. So my 3 organs that I wanted to study are the Leydig, of course, my spleen as a positive control because we know antigen presentation occurs there, and the heart as our negative control because it's a non-immune tissue. So we don't expect to see expression of these immune molecules in it. So now that I have my tissues, I want to degrade them and collect the RNA because RNA is the precursor to the proteins, and that's what I want to study. However, I can't look at just RNA. I need it in its DNA format. So I'm able to use an enzyme that allows me to go to cDNA, and then I'm able to use it as a template for further study. And so I reached the focus of my summer, which is primer design. So primers are part of a process called polymerase chain reaction. And what that allows me to do is to select very specific areas of a template and amplify it. That way I can study it. So I use 2 databases to be able to design my primers, Skatebase and Decipher. And Skatebase is the product of 5 institutions known as the Northeast Cyber Infrastructure Consortium. And Decipher, I was given access to it by Ben King here at MDIBL. So I want to take you quickly through the workflow of using one of these databases and how they can help us in terms of answering physiological and molecular questions when we're using bioinformatics. So let's say I want to make primers for TCR alpha, one of the genes I'm interested in. I just can't simply go online and type in TCR alpha little skate. It's not annotated. So I have to start with a reference gene. So what I want to choose is an animal that's closely related to the skate. So in many of my genes, I used the nurse shark, which is another elasmobranch, and I was able to find its TCR alpha sequence online. I can then take that sequence over to one of the databases, so Decipher, for example. And what Decipher allows me to do is take that sequence, that DNA sequence, and compare it to the genome and transcriptome of the skate. The result page will look something like this, and while it looks like a lot of nonsense, it's essentially just showing me what aligns between the information we put in, the nurse shark information, and what we get out is the skate sequence. So that's great. Now I know what the sequence of the skate is, and I can make primers for it. But I really need to make sure that I'm isolating the gene that I want and not some other piece of DNA that's not going to really work for me. So what I do is I take that skate sequence that I now retrieved from Decipher, and I do something called a reciprocal cross, which allows me to compare the sequence that I got for the skate to a known protein database of another organism. So in this case, we could just use the nurse shark all over again. And optimally, your first result, once you put it through the program, is TCR alpha. And then with that, you can be confident that you're constructing primers for the correct gene. So I went ahead and did my PCR. And when I get my products, I know the size of what I'm amplifying. So I could run that through gel electrophoresis to see if I can find and try to see if there's expression of any of these genes that I want to study. So in the Leydig organ, I was able to see some faint expression of the MHC2 and 2 beta, and we see some TCRs expressed as well. In the spleen, we can see some expression of MHC2 alpha and TCR gamma. This third gel shows some TCR beta expression in the Leydig organ, and all the DNA I used here were from experimental animals. So there's still a lot of different PCR runs that we would need to do. So I've taken this slide to show you what I've isolated and found so far. So I found RAG2 and TCR beta in the Leydig, and MHC1 beta, 2 alpha, and TCR gamma in the spleen. So what does this mean for my project and the future of it? So it's nice that I was able to find some of these genes, but of course I haven't found all of them. But seeing that I can find parts of the subunits, I mean, remember that these are 3D structures that are comprised of different pieces. The fact that I was able to find some piece of that receptor might indicate that that other piece is out there. I just need to go back to my workflow and edit something. And that's part of my future directions. I would like to isolate RNA from additional samples. That way I can reduce the randomness of our sampling. We could redesign primers. We could go back to the databases and maybe choose new primers or choose other regions that aligned. And the really nice method that optimally I wish I could have done was quantitative PCR, because that's what allows me to measure gene expression across different animals. And what I would do is compare the gene expression between experimental animals that are responding to a pathogen versus the control animals that were not given that pathogen. And lastly, we would like to do some gene annotation, because ultimately we really want to understand what are the most structurally important parts of these molecules in terms of antigen presentation and maybe other immune processes. So I'd like to thank Dr. Bram-Lutton for allowing me to work in his lab this summer and for his wisdom, Dr. Benjamin King for giving me access to Decipher, Morgan Bresnahan, Taylor Hirsch, Alex Dimmond, and Marina McKinnon were all students with Dr. Bram in the past, and they all helped me out with their research and their protocols. Chris Smith for providing a lot of reagents and instruments for my protocols. And I would like to thank NDI and NSF for funding for this project. And with that, I'd like to thank you for being here today, and I'll take any questions.
Any questions for Juan?
Yes, great presentation.
Thank you.
The question I have is, I understand that Collaborate was some of the first to generate antigen-related presentation bodies. The fundamental question I have is that what do you hope, if you could do all those things, what do you hope to elucidate in terms of understanding what you need to understand?
Right. So what method would I conduct in order to be able to find the answer to my question, and ultimately, what am I trying to answer? So what I'd like to do, again, is the qPCR step to really assess the gene expression, because just finding the genes in these Leydig gels that I ran isn't enough to say that antigen presentation is occurring there. I really need to measure the different expression across both immune tissues and non-immune tissues to make sure that it isn't just a baseline expression that's occurring, and really indicate that the Leydig is the site of antigen presentation.
Could you do that histologically as well? Like if you had antibodies to those protein components, then immunolocalized to Leydig, or are you back to the same problem in that even if it's just some baseline that's not good?
So, right, so if you did some type of antibody staining, if that could help, I guess you would run into a similar problem where if you do have just a baseline expression, you would see some staining, but I guess you would have to do a comparison between experimentals and controls to see if there's an increase in staining. That's a possibility as well. Yes.
Did you use the general primers? And so how could you distinguish between MHC1 and MHC2?
So the question is if I use degenerate primers and how I distinguish between the MHC subunits. So I did not use degenerate primers, or just regular ones that I created through the PrimerQuest tool of an online website. And I would distinguish between the two different subunits by using these reference genes. So I would try to find as best as I could, like major histocompatibility alpha chain. And it's hard, and some of these genes have different names. But what you want to do is try to find each subunit individually and then make primers for them.
Juan, what would you, if your hypotheses were supported and you were able to do the qPCR, what would the discoveries you make here about the Leydig organ escape tell you about evolution?
So, so the question of what would, if I find the answer to my question and antigen presentation does occur, what does that say about evolution? So that's a really good question. I think what it shows is, and I think I'll go back to like this idea of the structures, is that a lot of these animals, so the Leydig is unique to them, and it shows us that these molecules are evolutionarily conserved. And I didn't mention this in the presentation, but the Leydig is bone marrow-like. It's kind of equivalent to our bone marrow. So it could also help us understand our MHCs and TCRs interactions as well. So that's some of the promise there.